So today we're comparing steroid versus non-steroid hormone cascades, and some examples of this are progesterone, estrogen, testosterone for steroids. So these guys are nonpolar, which means that they can come right through the cell membrane and go into the cytoplasm. And once they get into the cytoplasm, they don't really do that much in there during the cascade, and when they're dealing with signaling, and they go right into the nucleus through the nuclear membrane, through little wells. And they connect to different protein receptors. So there's a hormone receptor complex and an intracellular receptor molecule. And when they connect to these, it helps with triggering the mRNA to go connect to the ribosome and start synthesizing the encoded protein. So next we have the non-steroid hormone cascade, and an example of this would be epinephrine. So when we're doing non-steroid hormone cascades, they don't actually go through the non-steroid hormone itself doesn't actually go through the signaling process. It sends a signal of itself, as you see by the yellow bead connecting to the ATP, into the cell for it, because it can't go through due to it being polar. And so it sends a signal through, and it connects to ATP energy source, adenylene cyclase. But the adenylene cyclase that connects to the ATP can't do it on its own, so it sends it to cyclic endosine monophosphate, which is also known as CAMP camp. So this camp promotes a series of reactions leading to the cellular changes associated with the hormone's action. So an example of this would be the protein kinases that are inactive going to the protein kinases that are active, then going to the substrate signals that are inactive, going to active substrate signals, which this leads to cellular changes. Some hormones signal all the way into the nucleus, some of them don't and just stay in the signaling process.